back into it. We have a really special presentation today about type 1 diabetes and APS type 1. One of the conditions that we know is really challenging for our patient group, and so we're very excited today to have Dr. Mark Anderson and Dr. Alessandra Fierabracci to be speaking with you. Dr. Mark Anderson is a professor and physician scientist at the University of California at San Francisco Diabetes Center and a recognized expert in the genetic underpinnings of autoimmune diseases and the control of immune tolerance, and a long friend and supporter of the APS Type 1 Foundation. And Dr. Alessandra Fierabracci has traveled all the way from Rome to be with us today. She has been a senior scientist and group leader at the Children's Hospital Bambino Gesù in Rome, Italy since 2001. We are so grateful that you made the trip from Italy and California. We bring you here together. So thank you so much. I'll pass it over to Dr. Fierabracci.
Thank you, Jennifer, for the introduction. First of all, I thanks for the invitation. I'm very honored to speak in this audience of patients, clinicians, and scientists all together. And I'm very honored to share with Professor Anderson this session, this special session on the management of type 1 diabetes in APS, a rare manifestation in a rare disease. Diabetes is known from very ancient times for the entity of its clinical manifestation: chronic hyperglycemia, polyuria, and polydipsia. Acute and chronic severe complications affecting several organs may appear during the disease course. Since 1,500 years before Christ, that in Weber's papyrus is a reference to polyuria, one of the main symptoms of the disorder. And later, Arrhenius from Cappadocia coined the term diabetes from the Greek diabanein, referring to the increased flux of urine. And later, the English physician Willis adds the term mellitus, referring to the sweet taste of urine. It's only in 1974, although the disease is known from very ancient times, that with the discovery of the islet cell antibodies, Gianfranco Bottasso, who was my mentor, discovered that type 1 diabetes is an autoimmune disease. You know that in our body there is an immune system which is a complex armamentarium of cells, lymphocytes, T lymphocytes, B lymphocytes, innate immune cells, organs, and tissues that protect ourselves against external agents, bacteria, viruses, and toxic agents. But it's in particular conditions of interaction between genetic background and environment that the immune system can exert an abnormal immune response against our own cells, in particular, in this case, the beta cells of the islet of Langerhans that produce insulin. And insulin is going to be lacking in the body, so we need to administer insulin to our patients. Type 1 diabetes is a multifactorial autoimmune disease with strong genetic components, and in both Europeans and North Americans of European ancestry, susceptibility loci were identified within the HLA complex, but also HLA alleles that confer strong protection. I wish to remind that type 1 diabetes can be isolated, but also often can be together with other manifestation, clinical, subclinical potential, which means only serological, of other organ and non-organ specific autoimmune diseases contributed to the autoimmune polyglandular syndrome. As you know, when it's associated with hypoparathyroidism, candidiasis, or Addison's disease, is part of autoimmune polyglandular syndrome type 1. When it's together with Addison's disease, is part of the PS2. When it's associated with autoimmune thyroiditis, autoimmune polyglandular syndrome type 3. When it is associated with any other autoimmune diseases, chronic atrophic gastritis, celiac disease, vitiligo, etc., and many others, is part of the autoimmune polyglandular syndrome type 4. These syndromes are related to genetic components, IRE gene for APS or HLA genes for the other autoimmune polyglandular syndrome classes. As you know, it is important to be able to predict the disease onset because an accurate prediction allowed to establish measures, treatments to be able to prevent the disease onset, because at the time of onset of disease there is a residual beta cell mass that is going to be attacked continuously by the immune system toward its continuous destruction. And nowadays we know that in early stage there are genetic markers that in very early stage allow the genetic prediction, and there are trials that are aiming to establish measure of prediction at this stage. Later appear autoantibodies against important proteins in the pathogenesis of the disease that are nowadays used as combination markers to predict the disease onset. After the discovery of islet cell antibodies, there are other main autoantigens that help to predict the disease onset in combination test: glutamic acid decarboxylase, isoform 64, IA204, ZN-T8, and insulin, among the others. And there are ongoing trials that try again to treat the immune system in this stage to avoid or delay the disease onset. Here I remind one of our studies I have collaborated with Corrado Betterle and Marco Cappa and other endocrinologists in Italy. This paper is published on 158 APS patients in Italy, the largest survey in the world, and others are going to be added. We have seen after this publication. We have also encouraged the foundation, following your model of an Italian association of patients led by one of our patients, Genovese, and trying to have a registry of all these patients. We have genotype all these patients among the different regions and found peculiar genotypes. For example, we were telling yesterday with Talarico, the R203X mutation in Sicily, the R139X mutation in Sardinia, the R257X mutation in northeast of Italy, A21V, which is of the Calabria south of Italy, and U78 among the others. And you can see on the left bottom of the slides that many manifestations can be present in the same patient, and as a consequence of following up these patients in many years, we were able to produce this data. In the same paper, there is a comparison of the incidence of type 1 diabetes that you see with the red line, marked with the red line, in the Italian population in comparison with 20 national cohorts published from 92 to 2018. And you can see that varies between 3 to 33%. In different series. It's not surprising that in the Finnish APS cohort, the prevalence of type 1 diabetes is elevated in respect to what is estimated in Eastern and Central European origin, population, Irish or Arab families, suggests that possibly there is a genetic predisposition to this incidence in APS. In Sardinia, so in a different south part of Europe, which is known to have an high incidence of autoimmunity and type 1 diabetes, it is surprising anyway that on 20 APS patients, only one patient developed type 1 diabetes. Here is a summary of the incidence of type 1 diabetes in the Italian APS population and the mean age of presentation. 1.3% of patients, APS patients, had isolated type 1 diabetes at the onset, but by the end of the follow-up, 8% approximately of patients developed type 1 diabetes. But at the onset, all affected patients were positive in the serum for the presence of the known autoantibodies specificity. And 28 of patients without type 1 diabetes were positive for these autoantibodies. However, during the follow-up, the annual incidence of type 1 diabetes was 1.1%. By excluding the Finnish prevalence, which is higher, the average age of type 1 diabetes in APS patients is 8% in all series overall, similar to that report in the Italian population, with a mean age of onset, which is 8.5 years. There are several studies, and here I'm going to summarize, and Professor Kamp has contributed to this investigation in the past years, that point out to the low predictive value of those specificity we mentioned before that strongly predict type 1 diabetes incidence toward type 1 diabetes development in APS patients. And very interestingly, Professor Kamp carried out this investigation in the bottom of the slides, comparing GAD autoantibodies reactivity in isolated type 1 diabetes, Stiffman syndrome, which is known to have GAD autoantibodies because the autoantigen is shared also with the brain, and autoimmune polyglandular syndrome, concluding that possibly GAD autoantibodies target different parts of the molecule, suggesting that also at the thymic level in perinatal age there are different mechanisms on tolerance regarding this particular GAD autoantigens. Alternative pathogenetic mechanism may expect to underline that the molecule is present to the immune system through in alternative ways, and even immunoprecipitation studies show that all sera immunoprecipitated GAD from rat islets lysates. However, serum autoantibodies from Stiffman and APS patients recognize the human GAD conformation on Western blot, while isolated type 1 diabetes sera did not, and APS and Stiffman syndrome sera inhibited the GAD enzymatic activity, but not the type 1 diabetic sera. This suggests, as final remark, that GAD autoantibodies specificity are an epiphenomenon of an autoimmune insulitis that does not lead to clinical disease manifestation. But there is another important discovery that a particular autoantigen, the 51KD pancreatic islet autoantigens, was discovered and found specifically in sera of diabetic patients within APS. And this is the L-amino acid decarboxylase. These antigens was unrelated to GAD because if the islet lysate is depleted with GAD, the amount of these autoantigens was not affected. And these cohort of patients, APS patients, did not show altered insulin response, suggesting that indeed there is an autoimmune response against the islet different from that that underline the classical insulitis in type 1 diabetes. And here a detail of the paper from Veloso, who discovered this finding, that show that sera from type 1 APS patients may have a different reactivity on pancreas and cerebellum slides, and on the bottom right side of the slide you see that APS sera indeed react with 51 KD autoantigen. The isolated type 1 diabetic patients had a serum that did not recognize this specificity. Several investigations were further carried out in the Swedish court, in the Scandinavian court, and indeed the presence of the 51 KD autoantibodies were detected in high percentage in APS patients, none of the isolated type 1 diabetes patients, nor in healthy controls. But what is the contribution to the development of type 1 diabetes in APS cohort remains to be discovered. Regarding genetic factors that may explain the low incidence of type 1 diabetes in APS, there were hypotheses. However, the type 1 diabetes was not associated with peculiar HRA gene mutation, and the haplotypes of HLA predisposing to type 1 diabetes in APS or protective were the same that in the type 1 diabetes general cohort. I open a parenthesis because in APS patients there were not found in early studies any peculiar association with human leukocyte antigen class 1 or class 2. Subsequently, there was some indication that the HLA A28 was more frequent in APS patients than in controls, or the HLA-A3 was more frequent in APS patients with ovarian failure. Therefore, the question remains open. Significant differences were not found by examining the HLA class 1 antigen in patients, 17 patients than in normal controls, and betterly found that class 2 were at increased frequency of DR3 and DR5. There is also to consider that also single mutation of the I gene, we suppose that can have an effect on the predisposition to single common autoimmune conditions. Therefore, it is interesting to explore I gene variants or sequence polymorphism in these individuals that do not develop APS, and this is also valid for parents of the patients that have also elevated levels of IgA and activated the lymphocytes. The insulin genes has been largely investigated for type 1 diabetes genetic predisposition overall, and the locus IDDM2 mapped to the mapping within the upstream region of the INS genes and corresponds to a mini-satellite polymorphism of variable number of tandem repeats, having a role in the ectopic expression of insulin in the thymus and induces susceptibility to type 1 diabetes. These are the main studies conducting for the genetic susceptibility to type 1 diabetes in a set population, and you see mentioned these INS when RT alleles. However, there are also studies to mention for type 1 diabetes discordant APS siblings that have the same RNA mutation. The type 1 diabetic affected siblings had the genotype, while the unaffected did not present the genotype, suggesting confirming the involvement of this locus in APS-associated type 1 diabetes. There are no convincing evidence of this association analysis of the higher insulin genes in Finnish diabetic patients. It's important, but however, to observe that a particular polymorphism of the insulin gene, we are not talking about the promoter, was significantly found associated with type 1 diabetes in the Finnish patient population. In conclusion, rarely type 1 diabetes occur in a percent with different frequency among the different ethnic groups. Type 1 diabetes in APS has, for sure, peculiar immunological feature, as shown by the properties of the diabetes-related autoantibodies. Can be considered a particular genotype of of type 1 diabetes when it occurs in APS that can have peculiar response to immunotherapeutic approach. This is something to be explored, and maybe with Professor Anderson we can continue this discussion also on the clinical therapeutic perspectives. Thank you for your attention.
All right, so as many of you know, I've been working on AIR and APS1 for many years, but another part of my life that you may not be aware of is I'm also a adult endocrinologist and diabetologist, and I actually run our diabetes center at UCSF. And so I thought a little bit about my talk today, and of course, you know, I could tell you about the ins and outs of managing someone with type 1 diabetes, pumps, multi-dose insulin, continuous glucose sensors, but you have me here, and you know, I thought maybe what I would do is share, you know, how we think about, you know, this really complicated problem of not only how autoimmunity happens in patients that are affected by APS1, but how autoimmunity happens in type 1 diabetics, and how I think APS1 has been an incredibly powerful model for unraveling all of this complexity. So I hope that when we get to the end of this, you get a sense of how I think about this. So I thought this was a great place to start, similar to my hey, let's just tell you about a patient that I saw more than 20 years ago when I was doing endocrine training at Mass General. And at that time, this was a 51-year-old woman, and you can see the list of many of the things that you guys are familiar with on her medical problem list and what age they arose. But at the age of 40, this particular woman developed type 1 diabetes. And it was very convincing it was autoimmune because she had the autoantibodies that we see in type 1 diabetics. And then this is just a little model slide of like summarizing, you know, a long period of work that's not only been done by our group, but many other groups about just trying to understand how the gene that's missing in APS1, the AIR gene, what it actually does, what's its job. And so AIR is this amazing what we call transcription link factor that operates in these cells in the thymus. And it's not T cells that are developing in the thymus, it's cells that the T cells are interacting with in the thymus, right? And so the cells that are interacting with the T cells, we give them a really complicated name. They're called medullary thymic epithelial cells. And that's just because they're located in the medulla, which is the central part of the thymus. And they have an epithelial-like, a skin-like characteristic to them. And that's where AIR is mainly expressed in our body, right? And so that's a major clue into what AIR might be doing. And then we figured out by knocking out the mouse gene and making a mouse version of APS1 and looking at these cells, that what AIR is doing in these cells is sort of this amazing job to help teach T cells who's you and who's not you, right? Self and non-self. And so what AIR does in those cells is it basically tells those cells to start turning on genes that normally shouldn't be on, right? And a great example of this is insulin, right? So insulin is mainly made in our pancreas and those special cells in the pancreatic islets that help control our glucose levels. Well, it turns out there's a small amount of insulin that's made in the thymus. And this has long been known, but how it happened was not known until APS1 came along, and then the AIR gene got cloned, and then we got to this point. And when you look in the cells that are missing AIR, at least in the mouse model, they don't make insulin anymore. And AIR not only turns on insulin, but turns on all these genes that are basically sort of like the same model as insulin. They're made in one place or a couple places in our body and nowhere else, right? And so if you think about this for a minute, in what the job the thymus has to do in teaching self and non-self, it needs to turn on all these genes that normally shouldn't be in a given cell, right? And so that's the idea, is it's turning on these genes to teach T cells who's you and not you. And so with that model in place, I just wanted to tell you little snippets of work that are all connected to type 1 diabetes at some level. So the first thing I wanted to tell you about, what about humans, right? You know, in the lab, the nerdy mouse immunologists do a lot of mouse work. And the reason that we do a lot of mouse work is immunology is really complicated. And the mice are a tool for us to really get into the weeds and study what's going on. And you see me bouncing back and forth between human and mouse, but there's some exciting developments now in what's going on in the laboratory with their ability to measure human cells like we've never been able to do before. And so what I want to tell you about here is we've actually started doing studies on the human thymus with a technique called single-cell RNA sequencing. Right? I'll show you a picture of that in a second. And this work was done by a combo at UCSF. Audrey Paranto, I think many of you met before, who's been at this meeting before, who's originally Canadian, now a professor with me in my department. And then Joey Batista, who was a surgeon at UCSF. And Joey was critical in this project because where can you get a human thymus, right? It's in the chest. I don't think anyone in this room is going to sign up for a study where we're going to say, hey, we're going to take your thymus out. It'll be okay, right? But it turns out, right, that when kids have cardiac surgery, right, the thymus is in the way. And believe it or not, and this happens every day, every weekday at the hospital when kids are getting cardiac surgery. Cardiac surgery opens the chest, and the first thing he does is cuts out the thymus and throws it in a bucket. Right? Amazing, right? Right? Throws it away. And so Joey was critical in us being able to get human thymus that was otherwise going to be thrown away. Okay? So then, what these guys did together, without getting into the gory details, is we're able to take the thymus apart and get it into a single, what we call a single cell suspension. So the cells are now floating in solution where they're all separated from each other. And then you can put the cells into a pipeline where each cell gets measured and each cell gets all of its mRNA sequenced, right? And the mRNA is the messenger RNA. It's telling you what genes are turned on in that cell, right? So imagine that in this experiment, one cell, we can measure thousands of genes that are turned on or not turned on in those cells, and it gives you the identity of the cells. So what you're looking at in this fancy, what Rorschach plot here, right? Each dot is a cell, right? And then we're separating the cells in a two-dimensional space based on do they look like each other, right? So you're taking all the genes that they have turned on, and if they're similar, they'll be close to each other. If they're different, they'll be far apart. And then for the business of today, we're only looking at these thymic epithelial cells that I told you about in the first slide. And so then it turns out that the cells that are making air in this plot here, how do I get a pointer? I'll just walk up to it here, hold on. So these red cells right here, these guys right here are the ones that are making the air gene. Oh, there we go. Beautiful, thank you. Right, so these the bright red cells are the ones that are making the air gene. All right, and so then, I'm just gonna jump to this, right? So then, Audrey did this incredible bioinformatic analysis of cells and just said, hey, which of these cells is actually making genes that patients make the autoantibodies that we measure in all of you? Which cells are making the targets of the autoantibodies? And that's what's over here. And spot on, it's the cells that are making air. It sort of validates what we thought was going on, but this is the primary data. And then in the purposes of type 1 diabetes, what about the cells that make insulin in the human thymus, right? Where do they land in this little plot? And spot on, they land exactly where the air is being expressed, right? So humans, right, make insulin in their thymus, and air controls its expression in the thymus, right? Pretty amazing, right? But here you are, this is human data, this is not mouse data anymore. And so then, you heard this from Alessandra already, now jumping to garden variety type 1 diabetes, right, which affects hundreds of thousands of kids every year in the United States, right? There's well over a couple of million people in the United States affected by this disease. And so we can do the genetics of common garden variety type 1 diabetes and measure genes that confer risk, right? And the second highest risk genetic variant maps to the insulin gene, right? So the question would be, well, why the insulin gene, right? It might be like how insulin works in our pancreas. Well, it turns out that we think the biology is actually how insulin's made in the thymus, right? So people that have the risk allele for type 1 diabetes in the population, this is a common variant. They actually make less insulin in their thymus. And we think that that's why they're predisposed to getting type 1 diabetes. And so APS1 and AIR help put the whole story together, make this finally making some sense. Okay, so then I wanted to tell you about some other work, getting down into the weeds, right? I'm a scientist, right? There's one thing to say, hey, insulin's in the thymus, it confers genetic risk. I want to see those T cells being selected by insulin. How do we do that, right? And so this work I'm going to tell you about real quickly here is done by a postdoc in my lab, Jennifer Bridge-Smith, in collaboration with a senior immunologist in Denver, John Kaplan. And this is a very busy slide. Don't worry about any of the major details here, but Jennifer did this amazing experiment where she went into a mouse model of type 1 diabetes called the NOD mouse. So the NOD mouse is this mouse we use in the laboratory, which spontaneously gets type 1 diabetes. And it looks like it's somewhat similar to what happens in humans, right? So T cells attack the islets, and kill them off, and the mice become diabetic. And a lot of the nuts and bolts of what's going on in the mouse are probably similar to what's human. And so it turns out to be a great testing ground of understanding type 1 diabetes. And so what Jennifer did is she said, well, hey, these insulin-specific T cells, well, how does AIR select them? And so she went in and did single-cell sequencing of T cells from an NOD mouse that was missing the AIR gene and an NOD mouse that had a normal AIR gene, cloned out a T cell that we call 405. So 405 is just the name of a specific T cell, and that T cell sees insulin. And the reason we need to do that is now that allows us to look in the thymus and see how that T cell gets selected. And so the key piece of data is down here on the bottom, is what we see in the AIR knockout is more of these T cells leak out of the thymus. So this is like our first way to actually visualize those T cells getting selected. And because they get out of the thymus, they then go into the pancreas and start attacking the islets, and they cause more damage to the islets in the pancreas. So now we can see it. Okay, so that's one thing, is like, we've proven that AIR is definitely selecting these cells. But here's an interesting angle to this that came out of this work. And so I really have to give a lot of credit to Dr. Kapler, who we collaborate with, is how do T cells see the insulin, right? And it's some of you probably are, aficionados of immunology, but T cells see antigens that they're specific for in a different way than antibodies do, right? So they see little peptides or fragments of a protein on the surface of another cell, right? And so getting down in the weeds about insulin in the NOD mouse, it turns out that all of the CD4 T cells that see insulin, for the most part, see a peptide that we call register 3, which is in the B chain of the insulin gene. And this little bold sequence is what those T cells are seeing. And in the normal NOD mouse, and this sequence is identical in humans, by the way, the 9th position of this peptide, there's an amino acid called an arginine there. And it turns out that that arginine is suboptimal for how it's being held on the cell surface by the antigen-presenting cells. And we figured out that if we change that arginine to a different amino acid, it makes the presentation of the insulin better or stronger, right? And so then, many of you have heard this amazing technology that's come along called CRISPR-Cas9, where we can do genetic editing, right? And we can do that in cells, but we can also do that in mice, right? And so we went into the NOD mouse, and all we did was alter with CRISPR-Cas9 this one arginine and changed the arginine to a E, a glutamic acid. And the reason that we did that is now we think insulin will get presented better, and maybe that'll get expressed in the thymus, get rid of T cells better, and that could protect against diabetes, okay? So we did the edit, we made mice, and I'll show you the real spectacular results here. And so the black is the incidence of diabetes in the wild-type control mice. So all the wild-type control mice get diabetes between 14 and 25 weeks of age. And the red line on the bottom are these mice that have this one mutation. None of them get diabetes. 100% protection, right? And so we're still working on the exact details of why. But remember, I had that tool I showed you that we can use to measure how T cells get selected in the thymus by insulin being present. And so we're just going back to the tool. And so the key piece of data now is to look at this column here with this 405 mouse. We're looking in the thymus, and then this is the 405 mouse crossed the mouse that's presenting insulin in the thymus better. And what you see is these insulin-specific T cells that are in this box here, they basically all disappear, right? And so by introducing this one single mutation, what we're doing is making the display of insulin better. T cells can see it, and now they're properly educated, and you no longer trigger diabetes. And part of the reason I'm showing you this whole thing, it's right down in the weeds with mouse, but if you think about it for a minute, if we're gonna start talking about how we're gonna fix APS1 or type 1 diabetes, there will be a day where we get to re-engineer those cells in the thymus. And this could be part of the re-engineering strategy, as we go in and not only fix the AIR gene, but fix the kind of insulin that's being made in the thymus, right? Okay, so in the last couple of minutes, I just wanted to tell you about some other work I think that's been presented at this meeting before about measuring antibodies, autoantibodies in patients with APS1. And almost everybody in this room knows that the autoantibodies really are useful in us trying to understand what's the specificity of the immune system when it's causing a problem, like when it causes type 1 diabetes. How do we know that insulin is the target? And part of the reason that we know insulin is the target is we use the antibodies from the patient. And the antibodies come, at least in part, from T cell help. So even though the problem in APS1 is selecting T cells, those self-reactive T cells go into our mature immune system, and they help drive B cells to make antibodies against what they're seeing. Right? We call that cognate help. And so the antibodies, though, are much, much easier to study in terms of figuring out what they're seeing, because antibodies can just bind to antigens in solution. Remember how T cells have to see it on another cell, and they get super complicated in trying to unravel what the T cells are seeing. So antibodies are super powerful. And so a number of years ago, I got into this fantastic collaboration at UCSF with my colleague Joe DeRisi. And many of you may not know Joe, but he is really just one of the most joyful scientists I've ever worked with in my life. He is the head of our Chan Zuckerberg BioHub at UCSF that's being funded by Mark Zuckerberg's Facebook fortune. Right, so Joe has essentially almost an unlimited budget to do crazy experiments, right? And so a number of years ago, he told me he was interested in autoimmunity, and we had this really productive discussion, and he built a platform for measuring autoantibodies where we could test them against the entire human coding proteome. So every gene in the body in one experiment could be tested. I'll show you a cartoon of this in a second. And that work ended up being so productive, we ended up co-mentoring a UCSF MD PhD student together, Sarah Vasquez, who was at the last get-together here. And so I just wanted to highlight their work and just say what a fantastic collaboration this is. So this is a little cartoon of what we're actually doing. And so this is part of it's a technique that's been around for a long, long time in molecular biology, where we use viruses called phage. And phage are viruses that infect bacteria, but phage can be used as a library that we call an expression library. So each phage, you can make them make a little piece of a protein, is the way to think about it, right? And it's on the surface of the phage. And now with engineering, we know the entire coding human genome, right? We know every gene and we know its sequence. We've gotten bioinformaticists to tile out the entire coding human proteome in fragments, right? So we have a limit on how long we can go, practically speaking. But we took these fragments that are overlapping in the entire human coding proteome, shoved it into these phage, and then with the phage, what we can do is then take antibodies from patients that don't have APS1, might have type 1 diabetes, some other autoimmune disease where they're perfectly normal, and then we incubate it with the library. And when the antibody's detecting what it might be detecting, we can pull that away and spin that down and precipitate those antibodies, and then sequence the phage, right? Because the phage have the DNA that's encoding the little fragment that they're holding. And now we can sequence DNA like nobody's business in these large DNA sequencers. And so then we send it off, and we basically turn this protein assay into a DNA measuring assay. And so this is incredibly, an incredibly powerful technique, and it turned out to also be so productive that we ended up collaborating with Mihalis here. And I'm sure there are many patients in this room that actually contributed to this work. So I just want to thank all of you for the collaboration. But what I'm showing you here is now using this technique on APS1 patients and normal patients, and the darker color means that the antibody was present in the patients. And this figure is just listing out genes or autoantibody specificities that we knew already APS1 patients had. This is like our positive control, and you can see quite nicely that there's more red dots on the left than there are on the right, okay? And so it doesn't detect every autoantibody in every patient, but it does pick them up. But then this is the amazing part. This is the bigger experiment. This is, well, what new things did we find? And so this is now looking at a whole host of things that were never described before in patients and each column is a patient, and then the controls are off on the right. And what you can see here is, like, you look right here on the figure, look at all of these patients that have antibodies to this antigen called RFX6. We never knew this before, right? And I'll show you a picture of this in a second. So again, in one experiment, we got all of this incredible information. And so I'll just jump here as like some new things that we learned by doing this that we didn't know about. Is we think that we found out an explanation as to why many people with APS1 get problems with their enamel, right? So what's going on there? Well, we think the enamel problem probably is an autoimmune disease, right? Not a well-studied autoimmune disease. But the reason that we think that this is an autoimmune disease is one of the hits we got in this experiment was against an enzyme called ACPT. And don't be distracted by the word testicular in there, right? Because the gene is also made in the testes, but it turns out that humans that are knocked out for this gene, where they are missing this gene, they have one clinical problem, enamel hypoplasia. Right? And so what this is telling us is that T cells in these patients are probably attacking this enzyme, and then patients that have T cells that are attacking this enzyme can't make enamel correctly, right? And I'll probably be proven correct by you at some level when you're in your clinical trial if enamel hypoplasia gets better with immune therapy. So this is the other one I wanted to mention real quickly is RFX6, right? We picked up RFX6 in this experiment. So it turns out that RFX6 is a transcription factor that's really interesting. And so it's a transcription factor that is made in the gut, but it's also made in pancreatic beta cells, right? And there are occasional rare humans that have mutations in RFX6, and those children are born with diabetes. It's not autoimmune diabetes, it's failure to make enough islets to keep things in check. But they have another problem, and that is they have intestinal dysfunction. And the reason why is RFX6 is a key transcription factor. This is a cell that kind of drives the cell identity. And what RFX6 does is it drives the cell identity of these cells called enteroendocrine cells in the gut. There's cells in the gut that make little hormones that then communicate with other organs like the gallbladder and the pancreas and the stomach. And it turns out that many of you know APS1 patients frequently have problems with intestinal dysfunction. They end up sometimes going and getting biopsies that you saw in Mihalis' talk, and when the biopsy is done, often what's missing are enteroendocrine cells. And so we went back to the data with Mihalis, and on the bottom here, all we did is take APS1 patients and segregate them into, did they have intestinal dysfunction or did they not have an intestinal dysfunction? And did they have or not have the RFX6 autoantibody? And what you can see is the RFX6 autoantibody, it's not perfect, but it does a good job in enriching for people that have intestinal dysfunction. So we found a biomarker now for kids that are probably getting affected by this. Now, I should say that this is not the only biomarker. My colleague Alla here has found other autoantibodies previously that are also expressed in these cells. But anyway, just a new opening of the door. And so with that, this tool that I'm telling you about, I think it's a very powerful tool. Again, it's not perfect. One of the things about it that also makes it really exciting is it's relatively inexpensive. Right, so now that we've built it out and we've, of course it used the Zuckerberg money to get the pipeline going, right? We've gotten the cost down to $5 a sample in terms of our cost, right? And so that's incredible, right? Then it gets to the point of, is it a good experiment or not? More than it's the experiment itself slowing us down. So with that, I just want to thank the people that contributed all this work I was sort of speeding through, but wanted to give you a flavor of like how we think about how APS1 can really teach us something about type 1 diabetes. And, and getting down in the weeds is part of the key on unlocking all of this. And so, again, I think APS1 has been incredible in giving us clues into how type 1 diabetes works. And what's happening in this room is going to help a lot of people outside this room that don't necessarily have APS1. And then finally, the key people I want to mention, Mihalis, my colleague Joe DeRisi, Sarah, and John Kapler for the work that I told you about today, and Jennifer. Thank you.
I so appreciate these talks. They're really important to our group. I'm mindful of the time, as we have lunch getting served. Do we have one or two questions?
My son has type 1 diabetes, and he got it at age 7.
Yep.
But it was very slow progressing, not like classic
Yep.
diabetes. And but now he's like full blown diabetes. I mean, he
Yep.
has been for years. But what is challenging for us in managing him is he takes the steroid paralysis.
Yeah, absolutely, yeah.
His absorption is difficult. But now with you mentioning the difference, that it's, do I have this right, it's attacking the insulin, I feel like maybe sometimes, like we can't do carb rations for him. Because it will, every day is different. Like sometimes he doesn't need extra insulin for what he eats for whatever reason. I wonder if the immune system is sometimes attacking that insulin more so than other times,
Yeah,
and the other way.
yeah, I get your
It's
question.
so
Well, this is a fundamental problem with APS1, is right, you've got not only the autoimmune disease where you're destroying the insulin producing cells, right? But you've got the autoimmune disease destroying the adrenal gland, right?
Yeah.
And messing up the gut, and how we absorb glucose, right? So you've got these multiple problems you're juggling. And so one of the big, I didn't have time to get into it, but one of the big problems in type 1 diabetes is as it unfolds and marches along, the cells in the pancreas, the pancreatic beta cells that make insulin, are not the most hardy cells in our body, right? They're not good at replacing themselves, right? And so this is an unfortunate thing about type 1 diabetes, is once they're gone, they don't come back, right? And so this is what's so frustrating, right? Is, basically your son has, almost complete insulin deficiency, and now you're trying to match the insulin with the metabolism of the day.
Yeah.
What he's eating, how he's exercising, what he's absorbing, right? What his adrenal gland is doing. I mean, you have my admiration. The only, I think snippet of good, good news immediately right now in the whole space of type 1 diabetes are these continuous glucose monitors. Right? At least in my experience in clinic, this is a big game changing for so many people, because one of the big challenges in managing type 1 diabetes is overdoing it, causing hypoglycemia, and these CGMs have done a great job of stopping that from happening.
Yeah, he's on
Right.
Dexcom.
Exactly. So for the moment, we gotta manage all of this. We'll get to a point where we're gonna fix probably not just the type 1 diabetes, but all the autoimmune diseases together. That's how this is gonna have to get fixed.
It just appears that his basal is so much higher
Oh, I
than
believe it.
the classic
Yep, and
diabetes.
that's because he's got these other problems.
Yeah.
Yeah.
Sir, do you have time for one more question?
Sure. Yeah, I'll hang out. People can come
Yeah,
out here and so.
thank you, and
Yeah.
Alessandra also,
Yeah.
if you don't mind asking.
So at the beginning of your talk, you mentioned there was the 40-year-old that developed type 1 at age 40.
Yeah.
And it seems like the general consensus is type 1 diabetes is rare in 80%.
Relatively, well, wait a minute. No, well,
There seems to
we-
be like a slow progression in a lot of us.
Yep.
Recently, and there's these new autoantibodies that we're noticing have, do you think it's more common than we had?
yeah, well, I mean, Alessandra did a beautiful job of, showing us the numbers, right? And Mihalis has shown the numbers too, and it looks like if you take all identified APS1 patients in total, right, the incidence of type 1 diabetes, roughly 10 to 20%? Am I saying, does that sound about right? There's not a perfect number, right, because the N is so, relatively small, but it's around 10 to 20%. But what's interesting, of course, is what about the autoantibodies in type 1 diabetes, right, particularly GAD, which is a common autoantibody. That is very frequent in APS1 patients, well over 50%, right? And so what that tells you is many people that have APS1, their immune system is doing something to their islets, just not killing them and causing clinical diabetes. But as time marches on, you get older, right, the risk is still there, and then it, pops up when you're 40 or 50. Right?
65.
Yeah, there you go.
65, yeah.
Right. Right.
It's not, so it's, again, really difficult to get the diagnosis.
Yeah.
Treatment,
Yeah, well,
because you don't present like a type 1 and you don't present like a type 2.
well, no. I will tell you, like in my own clinical practice, right? Because I'm an adult physician, and of course, because I work on type 1 diabetes, I have a whole cadre of patients that got they don't have APS1, but they got type 1 diabetes when they were 55, 60 years old. It can happen later in life. It's more rare, but it does happen. And then this is another interesting phenomenon, perhaps for more of a sidebar conversation, is when people get type 1 diabetes after the age of about 25, it's a different disease than when children get it, typically, right? So typically when children get it, it goes through a rapid course and kids are, dependent on insulin. Adults, they can go, especially after they've been diagnosed, they have the autoantibodies, they're insulin deficient at some level, they can be on tiny doses of insulin for years, right? So it's a slower progression in adults. We don't understand exactly why that is, but it's an interesting phenomenon with